would like to start by saying we're incredibly privileged today to have with us Dr. Mossman, who is a member of the Groot Institute for Infectious Disease Research and the McMaster Immunology Research Center. Now, she completed her PhD in biochemistry and a postdoc fellowship in molecular virology from my alma mater, the U of A. Although I only did my master's there, so I can't claim the high level status from the U of A. She is currently appointed to the Department of Medicine and is an associate member in the Department of Biochemistry and Biomedical Sciences at McMaster. Her work, really, her seminal contributions are around cells sensing infection and how viruses escape being sensed or detected, which in the present day is incredibly topical, if you can imagine. But more recently, no buts, and more recently, she has been looking into how to use viruses to help attack cancer, which, you know, is huge, and the potential there has huge benefits to all of society. So I'm very, very, very happy, and I think we're all very privileged to hear from Dr. Mossman about her work, her ideas, and the potential impact of where she thinks things are going in terms of her research in the post-COVID climate. And so I just want to mention the structure today is going to be relatively informal. Dr. Mossman will talk for about 20 minutes or so, and feel free to chat and ask questions as you go. I will try and do my best to keep up. After that, we'll just have a question and answer and discussion, and bring up stuff between us, because I know some of you are experts in your own rights in related fields, so it'd be nice to have a good conversation. So with that, Dr. Mossman.
Well, thanks so much, and it's I'm really pleased to be with you today. It is I don't get a chance to think a lot about science anymore, so this is a lot of fun for me now that I've moved into administration. I don't get as much time to just think about science, and I have great people in my lab, and so I live very vicariously through all of my students and postdocs. So I'm really excited to speak with you today. I am going to share my screen. Here we go. There you can see that. Perfect. And so again, if you want to ask questions, please feel free to ask questions during, or we can have a conversation afterwards. So for many years, probably at least the full 20 years I've been at McMaster, but even before that, I've been really interested in virus-host interactions, and particularly with herpes viruses, although we now work on a whole number of viruses, including currently SARS-CoV-2. But there's a reason for working with herpes viruses. So Darwin, in his seminal work, The Origin of the Species, speculated that herpes viruses are likely one of the oldest, if not the oldest, of human viruses. And the rationale for this is because they have evolved to persist within the host for the lifetime of a host. So that's an extreme example of evolution between a virus and its host. And so as such, they're fascinating viruses to study because they can not only teach you a lot about viruses and virus replication, but they also teach you a lot about the host immune response and aspects of cellular processes. So this is a picture of a herpes simplex virus type 1. It was taken when I was a postdoctoral fellow, so you can get an idea of what the viruses look like. And the question really that I've been interested in for a long time is how can we use this sort of basic or fundamental information on virus-host interactions and how they modulate and evolve with a host immune response? And how can we use that information to develop anti-cancer immunotherapies? And it's, in some ways, it's quite simplistic. Of course, nothing is this simplistic, but the theory or the general rationale is quite simplistic. And it's, for example, if we take interferon, which is both an antiviral cytokine, but also an anti-tumor or an anti-proliferative cytokine, because interferon, one of its functions is to control cell growth, many cancer cells have mutations somewhere in the interferon pathway. So normal cells with functional interferon, there are limits on cell cycle and apoptosis, but in cells with non-functional interferon or in cancer cells, these sort of restrictions no longer exist, or there are mutations throughout that pathway. And because interferon, its other function is to block virus growth, many viruses can now replicate in cancer cells where they would be restricted from replicating in normal cells. And beyond interferon, you could really substitute interferon for the word apoptosis or cell cycle control pathways. But basically, fundamentally, pathways that control normal cellular growth are also ones that control viral infections. And so this is what establishes this whole concept of oncolytic viruses, which are viruses that preferentially replicate and kill tumor cells. So during the tumorigenesis process, it's like cells are doing a deal with the devil. They gain immortality and they gain continued cell growth, but this is really at the expense of protection against a viral infection. Now
Sorry to interrupt. Is there any way that you could just swap displays at the top? Because I think we're seeing your presenter view as opposed to the full slides.
Oh. I don't know.
It should be right up above in the little arrows there.
Is that better?
Thank you very much.
Oh, perfect.
Sorry.
No problem. So in the early years of oncolytic viruses, what was really thought is that, and this is how they got their name, that they're onco and they're lytic. So, they function by killing or lysing cells. And so if you allow the viruses to replicate uncontrollably in tumor cells, that's how they'll get rid of the cells and they'll control a tumor. And so early on, oncolytic viruses were tested in combination with drugs that were actually to inhibit the immune response. And so they thought if you, at the time of delivering an oncolytic virus, you also inhibited the immune response, you would allow the virus to continue until there were no more cancer cells left for the virus to kill. And then therefore the virus would run out of host cells and the tumor would be gone. And that failed miserably. So when that was tried in preclinical and clinical models, failed miserably. And so now we know that the lytic part, the actual physical killing of a tumor cell, only plays a small role. And it depends on the virus, nothing is absolute, but for a lot of viruses, it actually only plays a small role. And actually it's that induction of an immune response or the ability of a virus to stimulate an immune response that really is a large part of how these viruses work. And so when you think about immunotherapy, and everyone has now heard about immunotherapy, there are so many different types of immunotherapies that are out in the clinic, some really successfully, some still to be their full potential realized. But two real general aspects for successful immunotherapy is you need to have what we call an inflamed tumor or a hot tumor, where you have an infiltration of immune cells. But then you need to sustain that activity, because we know tumors can evolve. Tumors can respond, and tumors can evolve adaptive mechanisms to shut down this anti-tumor immune response. So really great immunotherapy approaches not only drive a hot tumor, but they also help to sustain that activity. So when you think about the cold tumors versus hot tumors, and that has to do predominantly with T-cell infiltrates, although we're learning about other immune cell types that play a role, you can see where different tumor types have a range of generally being thought of as cold. And so you see rhabdoid tumors, thyroid, AML on the cold side, and then those that tend to have a very high mutation rate being on the hot side. So, or with large T cell infiltrates. And again, when you look at mutation rate, it's not absolute. There's a general trend. But mutations where melanoma due to UV radiation, lung cancer in large part due to toxicity associated with cigarette smoking and other types of toxicities, you can really see that mutation rate in general does correlate with the T-cell infiltrate. But you can also see there's a vast range over a number of tumor types. So one of the challenges with using HSV1 is that, well, to back up a little bit, one of the first, and actually one of the first viruses that were really shown to be used as an oncolytic in preclinical and then clinical models was HSV1. And so even though there were anecdotal evidence of the individuals that had a tumor, they'd get a viral infection, the tumor would go away. When they actually started testing this in preclinical models and in the lab, the first real studies were done by a group in Harvard and based on HSV1 or herpes simplex virus type 1. And so we also started working with HSV1. It's the virus I was working on during my postdoc. And we already knew, and I was working with mutants of this virus, which are deficient in ICP0, because we know ICP0 is one of the mechanisms that the virus uses to counteract interferon. And so I told you before that many tumor cells are deficient in type 1 interferon. And so I had been really interested in just that virus-host interaction and really looking at what role does ICP0 play in the context of HSV1 in counteracting the effects of interferon. And so before oncolytic viruses were truly sort of established, particularly in Canada and really understood, and it's John Bell's group in Ottawa that have been leaders in this field, we knew that ICP0 deficient viruses grew very poorly, if at all, in normal human fibroblasts. And that's where we were studying a lot of their effects. But I propagated this virus and made high titer virus stocks on an osteosarcoma cell line. And so when John Bell's work came out, in the early 2000s, really looking at a totally different virus, VSV, and showing that VSV, it also has to do with this interferon response and the ability of tumors that can't respond or can't make interferon to provide this nice niche for replication of mutant VSV viruses. We started putting two and two together and realizing, oh, here we have a virus that can't grow in normal cells, but we propagate it in tumor cells. And is this unusual, or is there something to this? And so this is really the first model where we used an ICP0 null HSV1. And what we're looking at here is fold change. This isn't a breast cancer model. We've done many, many models since then. But as you can see in the dark colored, either with a single treatment or if we used basically killed virus, here we use heat inactivated or heat shocked virus, so dead virus, the tumors continue to grow and eventually you have to sacrifice all of the mice. Whereas if you use replicating virus, and here we use again 3 doses early on, we can see tumor regression and about 80% survival of the mice. One of the problems though with HSV-1 is that there's pre-existing immunity in the population. So depending geographically where you're located, up to 90 to 95% of the population is infected with HSV-1. And so pre-existing neutralizing antibodies. And one of the holy grails now is to be able to deliver either directly into the tumor or to deliver systemically. Deliver systemically to reach hard to reach sites, to reach metastatic lesions, and to reach, potentially tumors that are still very nascent and you don't pick it up on a scan. So that's somewhat of the holy grail. And we knew HSV-1 would be really challenging to use because of the pre-existing immunity. Also within this space, and this is where the logistics comes in, it was almost impossible to get IP because the very, very early IP that, when colleagues were starting to do these studies, many, many years ago, they were basically able to get IP that's, generally states HSV-1 with any manipulation for any type of cancer. So the field and the ability to get IP became really restrictive. And so even though it's a great model for us to use, and we use it, and we use HSV mutants because of their ease of use to get into the mechanism, for moving forward clinically, there were a number of reasons why it wasn't an optimal virus. So we took the stance of using something very, very similar, but different, and that was bovine herpes virus type 1. So BHV1 is in the same subfamily of viruses as HSV1, so in many ways very similar, but it is exquisitely species specific to cattle. And so it's what causes shipping fever in cattle. So when cattle go to the market and they get very stressed, they can get shipping fever. And so individuals that work in the cattle industry and at abattoirs and in shipping are really highly exposed. And there's never been any known seroconversion or any disease profile at all, even for individuals that have been really highly exposed to this virus. So we thought that this might be a good virus to study. So we decided to test the NCI 60 cancer panel. As many of you know, in the cancer field, this is a standardized panel. Many, many, many new drugs and therapies are tested on the NCI 60 panel. And so what we did is we engineered our BHV1 to express GFP, or green fluorescence protein. And then we looked at, when I say replication, this was increasing amounts of virus. So starting off the very, very low amount of virus and increasing, and we're really measuring fluorescence from GFP. And then we looked at cell death. And unlike our HSV-1 counterpart, that we saw, effects on maybe 20 to 30% of the NCI 60 panel. Here I'm showing you what we saw with BHV1. In that closer to 80, 85% of the NCI 60 panel, we saw fairly significant levels of cell death, even at low MOIs, and also under conditions where we really don't see evidence of replication, which really brought into question that whole concept of the lytic aspects of BHV versus, the actual replication. And we've since done quite a bit of work, and a postdoctoral fellow in my lab has actually published a review paper on, our understanding, and others have seen this in other circumstances where it's really unclear if you even need a productive infection at all. And I think it will be virus specific, but it really now draws even more into question, how are these oncolytic viruses really working? But this virus has become a really unique and interesting virus for us. So what are the pros and the cons of using oncolytic virus? Well, we know they're very good at inducing an immune response. So even in cold tumors, they're really good at inducing an immune response. And the beauty of an oncolytic virus is, unlike other therapeutics, it doesn't matter what type of tumor it is necessarily. It doesn't matter what the receptor status is. It doesn't necessarily matter what the mutational status is. Viruses are really good at getting into most cell types, and they're pretty good at stimulating an immune response. The downside to that is they're inefficient on their own. So very, very few oncolytic viruses, even if they work well in a murine model, and again, you know, you could probably set up any murine model to get the results that you really want. But when it went into phase 1 clinical trials and into the clinic, we really came to recognize pretty quickly that a virus, an oncolytic virus in and of itself, is not sufficient. We also found that it's really insufficient to overcome this tumor adaptation, and in part because you really want the virus to stimulate the immune response, and you want an antiviral response, but more particularly an anti-tumor response. But if the virus is really good at stimulating an immune response, the immune response is also going to be really, really good at clearing the virus. So it doesn't, you now have a great sort of immune response, but the tumor can adapt, and so the virus in and of itself as an oncolytic virus is not very strong at overcoming this tumor adaptation. But what we and others have found is that they can really synergize with other therapies, such as checkpoint blockade immunotherapy. So most people know about checkpoint blockade, great immunotherapies that, you know, target, you know, T cells and really allow for the maintenance of the activity of T cells. Immunotherapies, these sort of immunotherapies, checkpoint blockade in the clinic, work really, really well in those hot tumors. So we see really good efficacy of checkpoint, but only in very specific tumor types. And those seem to be the types of tumors with a really high mutational burden that already are inflamed or hot. And so this is where oncolytic viruses come in that they can make a cold tumor hot, and then the checkpoint can help sustain that activity. And so this is starting to go. So here's an example of what I just talked about. So this is with our BHV1, and these are, you know, quite challenging melanoma tumors. And so with just PBS alone, and this is looking at CD8 T cells, these tumors do, without any sort of therapy, have some T cells. They're pretty much, this particular model is pretty much what I would call a cool tumor. They're not totally cold. You do see some T cells. They tend to be at the periphery, not so much in the core. Checkpoint alone increases the amount of T cells a little bit, but not substantially, and certainly not to have a statistical effect on survival. If you have BHV1 alone with checkpoint, again, we do see an increase above PBS or even just BHV alone, not really sustained. But it's really now when we have what we call a triple therapy, and this is what we're trying to understand, where we use low dose chemotherapy and really low dose. So no cytotoxic effects of the chemotherapy. So chemotherapy alone, and here it's mitoxantrone or mitomycin C. I think this is mitomycin C, but very low dose chemotherapy, which again alone has no efficacy, but seems to really synergize with the virus. And then you add checkpoint, and we see quite a substantial increase in CD8 cells, both at the periphery and within the core. And when you look at percent survival, it's this, what we call triple combination therapy, that really allows that increase in survival, and where we really start to see efficacy. And this is what they're also seeing in the clinic. So for example, the only oncolytic virus to date that has been FDA approved is an HSV1 called T-VEC. T-VEC on its own wasn't actually just the virus, it also expressed GM-CSF. So it was already a dual therapy. And even though it was approved, it's not very good. And so it's not really used extensively. It was only approved for melanoma because with HSV, again, you can't go systemic. You have to be able to inject into the tumor. So it's really only used intratumorally. And it does have some efficacy, but on its own, even with GM-CSF, it's not an overwhelming positive therapy. There are clinical trials now using TVEC in combination with checkpoint blockade, and those clinical trials are showing really, really exciting data, and in a couple of other models, but still using intratumoral. We don't really understand, and this is where we're starting to go back and do those fundamental studies, RNA-seq and other types of studies to really understand how the virus synergizes with low dose chemotherapy. And we're starting to get clues on what those synergistic effects are, because of course, to move forward in the clinic, we don't want it to be a triple therapy, even if it's a dual therapy. How can we now modulate the virus so that we now only have a dual therapy instead of a triple therapy, like how they inserted GM-CSF into HSV-1. So what are the opportunities and challenges? So we're currently working with Novatio Innovations to move this forward. The opportunities, certainly the success of immunotherapy. I mean, immunotherapy in general is really showing a lot of efficacy depending on the tumor. It's also a little bit more of sort of a personalized type. The whole point of immunotherapy is you really get the power of the individual's immune system working for them. In the case of an oncolytic virus, the virus is long gone, but the effect of the immune response is still there. And so we certainly see safety with oncolytic viruses in clinical trials, been shown to be, really, really safe, unlike a number of the conventional sort of more cytotoxic, where there's a lot of side effects, very few side effects with oncolytic viruses. And that's in part because they're really specific for tumor cells, and they're restricted from growing and having any sort of deleterious effect in normal cells. As I mentioned, TVEC on its own, even though it isn't, a really effective oncolytic, for us in the field, it's been huge because it's paved the way for a whole new type of therapeutic modality to get into the clinic. And the more we all learn and the more we get, more oncolytic viruses in clinical trials, the more we're understanding that even though TVEC on its own doesn't show huge efficacy, now in combination with checkpoint and others, we're starting to see where I think it really can move the needle. So that's really exciting. And TVEC is really, because if you think about it, there are a lot of hurdles for TVEC to overcome. You're asking the regulatory bodies to allow you to stick a replicating virus into a really sick cancer patient. Right. And what we do know as well is, once you go into try to get into a clinical trial, you have to allow the patients to undergo, approved therapy. So the types of individuals you can enroll initially into these clinical trials tend to be patients that have really exhausted all other therapeutic options. And so, the first, probably 5 or 10 years of phase 1 and phase 2 clinical trials with oncolytic viruses were really disappointing. And for many years, the field was very, very deflated. But then once we could prove that safety and we're allowed to use higher doses and go into patients that hadn't necessarily exhausted all other therapeutic options, now there are many different OVs in clinical trials, mostly in combination with checkpoint blockade. So what have the challenges been? Well, as I mentioned, the limited efficacy of TVEC as a standalone. So the therapeutics are getting a little bit more complex, but again, that's not necessarily unique to oncolytic viruses. Many different tumor types, a single therapy is not the same. It's usually cocktails of therapies. And so the fact that, oncolytic viruses will have to go into the clinic along with other modalities such as checkpoint, that's not unique, I don't think. If you think about any other type of cancer therapy, it's typically a cocktail. But that does make it more challenging. Certainly, manufacturing and production of large scale is a challenge with oncolytic viruses. Depending on the virus, it can be very challenging and very expensive to manufacture in the type of doses that you need for these types of experiments. So manufacturing and production is always a challenge, which you don't really think about when you're doing, your preclinical studies and you're only going into mice. But it does become a fairly significant hurdle when you get to the point of wanting to move into a clinical trial. Right now, there really is, I would almost say, oversaturation of immunotherapy and clinical trials. One of the last meetings I was at pre-pandemic, when we actually met in person, someone had showed, I don't know where they got the information, but showed an overview of all of the current immunotherapy trials. And it was in the thousands and thousands and thousands of all different types of different modalities. And so you really need to have something that is really unique and different, that has the potential to really offer something new. Within the oncolytic field, we're discovering many new qualities of BHV1. So far, the one that we're the most excited about is not even just the ability as a large virus to be able to incorporate multiple therapeutic transgenes and really to be able to manipulate it that way, is the fact that we don't seem to need replication, which we don't understand. That sort of puts the whole oncolytic on its head, but it seems to work really, really well. And actually, the wimpier the virus is in vivo, the better it seems with its efficacy. So again, we're really trying to understand that, but that increases its safety profile. And all of our studies so far are pointing to the fact that we can use this virus either intratumoral or systemic. And the fact that, individuals don't have immunity or any sort of natural immunity to BHV1 gives it a huge advantage over all of the other human-based viruses where there is pre-existing immunity. The biggest challenge for us has been that even though BHV1 can enter into and replicate really well in human tumor cells, as you saw with the NCI-60 panel, it can't get into mouse cells. So this has been really challenging in doing preclinical studies. We did do some initial preclinical studies in cotton rats. They're susceptible to BHV1. If anyone's ever worked with a cotton rat, I had one PhD student who was willing to do it. And then once she got her paper and some really nice data, she said, find someone else. You have to wear Kevlar gloves. They bite, they jump. You can only house them individually. They're really, really challenging. It's a great model, but they're really challenging to work with. So we've had to rely on transgenic models or engineering murine tumor lines so that they're susceptible and they will allow the virus to bind and enter. So, there's always some challenge. For us, it's been the animal model that's really been a challenge, but we've again figured out ways of getting beyond that. So we're now in working with Novatio, really getting all of the as much of the data, that the preclinical data we can, to put a package together, and we're really excited to move this technology forward. These are the people in the lab. The ones that have really done the most work on this particular project moving it forward are Susan Collins, who's in the top right, and Maria Davola, who is just below me. They've been really the ones that have been driving this particular project. And with that, I will stop sharing.
Wow. So first of all, thank you so much. It's overwhelming to think of all the potential and the possibilities. I mean, first of all, if you are able to get the efficacy up, and some of the questions in the comments were in the chat included things like, well, can you use CRISPR to alter the virus somehow to make it more effective in signaling? Some of the other questions involved looking at, why is it that the earlier successes didn't translate into clinical studies? That's another point of discussion. And another question that was interesting is the efficacy consistent throughout all stages of a tumor's cycle? So early stages, is the virus maybe less or more effective than later stages? That's another interesting question. But I'd like to actually start with a little bit more broadly. You've mentioned the company you're working with several times, I think, but not quite in specific. So how has the industry, why did you partner? Why are you working with the other side, with the horrible, medical industrial complex?
So we've been really fortunate. We're working with Novatio Innovations, and they're, they really help assist with the stage that we're at. And they've been really great, incredible partners. They have, individuals on the team that have been in the oncolytic virus field, and have experience in that field. So it's actually been incredibly helpful to understand, because there's a big difference between the types of experiments we would normally do in the lab as, to understand mechanism and as an academic researcher, versus the types of experiments that you really need either to get, Health Canada or FDA approval to get your experiments into a clinical trial, to get the investment. Because you can't go into a clinical trial with an oncolytic virus on a CHR grant. So working with, with this group in particular that really understands both the biology, but understands the field and the regulatory aspects and the investment, has been incredibly helpful, because we've needed to set of experiments that you would not necessarily do or get CHR funding for, because it's not necessarily hypothesis driven.
Oh my God! So I think I'm shocked and dismayed to have an academic researcher talk about collaboration with the dark side, and things like funding. Oh my God! And understanding and collaboration towards clinical trials. Where is this all coming from? Is this, I'm just thinking about where you also sit in terms of your administrative work. How can you sanction this? This is horrible.
No, this is awesome. I mean, it's, to me, it's, and this is, now I'm going to put my VPR hat on, my Vice President Research hat on. I think what we do at an academic institution is generate, fundamental knowledge. But if you're not then working to put it into the hands of people who can use it best, and that could be in a commercial sense, in a social innovation sense, for us to generate this huge amount of knowledge in whatever discipline and not get it into the hands of people that can use it the best, then I think we're doing a disservice, not just to our funders, but to the community at large. And the one thing I recognized is, I have a lot of skills, but being a CEO of a company is not one of them. And it's not something I want to learn. So I'm more than happy to work with others that we can get it to a certain point, but they can take it and run, because I've learned it's a completely different skill set. And I don't have the time, the energy, or even the desire to learn that skill set.
So I guess the next thing I want to ask, and the next obvious question, at least to me, is that I gather that at McMaster's, there's a lineup of these people from industry standing at your door, knocking all the time. Pick us, right? There must be some magic formula that I'm unaware of, but because the collaboration with industry, how does that done? How did you do it?
So we've been at this for a very long time. We had been working with another group early in the process. Unfortunately, the group we worked with had not worked with anyone as early stage and as sort of at the fundamental research stage as we were at. They had only ever, worked on technologies that were already somewhat to market or within a phase one. So that became an incredibly challenging partnership because they didn't understand that, you can have a timeline and you can set milestones, but not everything goes the way you want it to go. And not every experiment works. And, that's the nature of research. And they had never worked within a really early stage. So expectations on both sides became inherently challenging, and we actually had to break off that partnership because we were at two different fundamental stages with two different sets of expectations. So it doesn't come easier overnight. You do have to find the right partner, that's really invested in the type of research and technology that you're doing. And we're just really fortunate that we found that. But, we work really closely with with Milo, which is our innovation or our industry liaison office. And again, they have a lot of expertise that, we don't have in the lab, but help set up these partnerships.
So I really like your point about the idea that, not the idea, the fact that there are different skill sets and different metrics and different endpoints between academe and academics and industry scientists, even, who used to be at one point academics, but have had to be acculturated into a slightly different way of thinking. And yet they can now, because they have new bosses, they can't turn back and say, oh, don't worry about it. It's okay if this fails. The millions of dollars that were spent, that's all for the good of basic knowledge. So that's an incredible gap. One other gap I noticed that I want to pick up on, and I welcome people, if they want to ask a question directly, please just put up your hand, and then we'll acknowledge you and turn on your mic. But one of the other things that I was very, very sensitive to when you were talking was the limitation for research with respect to production and manufacturing. So I know that currently the Canadian government is very into this notion of let's increase our biomanufacturing capacity. Are you aware of, or have you heard of any potential collaboration between government and industry to provide manufacturing capacity for clinical trials and research to make that bit easier?
Yeah, we're having, lots of conversations now. Certainly the pandemic has spurned those conversations. So we're fortunate at McMaster. We have a GMP facility to make viral vectors. It's been operational for 17 years, but we don't get a lot of government funding for it at all. It's hard to sustain it. It's quite expensive. In many cases, it's small scale, so it might be sufficient to get sufficient viral vectors for maybe phase one, but not for phase two and phase three. And so part of the conversations that we're having now is, how do you generate, and there's a number of these facilities across Canada, but they've never been coordinated, for one. So how do you coordinate all of these facilities to generate an entire ecosystem that during a pandemic you can quickly mobilize and use these facilities, including the one in Montreal that, the announcement just came out last week that this big, huge vaccine manufacturing facility in Montreal has now been built, but they don't have the nimbleness to say, do some R&D in small scale. So how can you use, these consortium of facilities that do exist, plus they need to be augmented so that during a pandemic, you have the capacity, but off cycle, you can use them for these other modalities, be it cancer therapeutics or other vaccines or other types of gene therapy, so that all of the investments that are made are used and leveraged strategically. So, those are the conversations that we're currently having.
Okay. Jamie, you had a question?
Yeah, well, I mean, just sort of on the topic of, I worked at Sanofi Pasteur, so I've had some time in the vaccine industry. And given the sort of real versatility of these viruses, is there a potential or interest in the development of a vaccine of a modified version of these oncolytic viruses that could be maintained at a stable, low level as a sort of prophylactic measure against these cancers, given that, you mentioned that the type of cancer per se doesn't really matter so much. They seem to be quite effective in most types of cancers. Or given the fact that, it has failed a couple times, is this a moot exercise until we perfect the science of these oncolytic viruses?
So that's a great question. And so there are different strategies that people are using with oncolytics. So one is, and this is what Turnstone Biologics, which came out of both McMaster and John Bell's group, they're actually doing a Primeboost strategy. So it's like a vaccination, right? It's a Primeboost. The advantage of that is you can get really, really high immune responses because you're boosting like you would with a vaccine. The challenge is you have to know, what are you boosting against, right? So you have to know what the tumor is, what's the major, immunogenic tumor antigen. And so when you know that, it's incredibly powerful, but then it's limited, right? Whereas the approach that we're taking is more, it's not as robust because it's not like a Primeboost, but it's sort of tumor antigen agnostic, but it will generate an immune response against that particular tumor. And I noticed one of the questions, and just really quickly looking, is, does it matter? And I think you mentioned it as well. Does it matter what stage? I mean, it is that the challenge is that tumors always are adapting, mutating. When they go to a secondary site and form a metastasis, they often change their characteristics and change their major tumor antigens. And so, I think that's a challenge. Even if you do induce a really robust immune response, it's not necessarily directed against, the fulsome nature, but there's a lot of work also trying to understand how can you manipulate the viruses and what they do so that they're generating a response against multiple different antigens. And we're starting to learn the sort of the immunology behind that.
Perhaps there's also, thank you, Jamie. Perhaps maybe there's also an opportunity to think about it from a precision medicine perspective. Rather, one focus is to absolutely improve the efficacy of the virus and the specificity, perhaps, or generalizability of the intervention. But maybe the other is to be able to administer tests to test for specific potentials for specific cancers that individuals have. The BRCA1, BRCA2 gene that people were very much into looking into. Maybe there are histories that people have that would suggest they should be tested for types of cancers, and if those genes are in their genetic makeup, then they go to this intervention, which is going to be highly effective as a primer boost. Has that been something that people have thought about?
So there's two different parts there. So one, if you look at mutational status, some oncolytic viruses, just depending on their nature and their virus-host interactions, preferentially replicate in tumors with specific mutation profiles. So that's, you need something to differentiate a tumor cell from a normal healthy cell. And so, for some oncolytic viruses, particularly the human viruses that you need to mutate to get that therapeutic index between normal and tumor. The viruses are being mutated really to take advantage of specific mutational profiles of tumors. So we already have some oncolytic viruses that preferentially replicate in tumors, for example, with a RAS mutation, or preferentially, mutate in tumors that have a p53 mutation, or certain types of mutations, and the viruses are generated based on that mutational status. When it comes to the immune response and generating the immune response, that's not predicated so much on the mutational status. It's more on what is that immunogenic antigen that the immune system will react against. Sometimes it's a mutated peptide. Sometimes it's a normal protein that's just overexpressed under certain cancerous conditions, but it's not always a mutated peptide. Sometimes it's just, a certain protein that's normally expressed at really, really low levels is now overexpressed. And there's only certain, and this is where we're starting to understand what are those immunogenic peptides that really will help stimulate that anti-tumor immune response. So there's two different, but there's certainly within the field, looking at all of the various strategies to either target the mutational status versus, or in addition to that sort of immunogenic peptide perspective.
So I think we have time for one more quick question if anybody wants to. Otherwise, let me give people a chance. Is there anyone that wants to jump in? Okay, seeing none. Dr. Mossman, I would very much like to thank you for talking about your work and your research. And I think that for our community and for the people that join us, both in the summer on a Friday afternoon, but also throughout the year, thinking about different kinds of research and the potentials and bringing people together around these kinds of discussions is incredibly important. It opens our eyes up to the various directions that science is taking and the various opportunities that I think we have for collaboration and for translation in the near future. For one, your particular work is by no means my area of expertise, having graduated with a English literature degree, from the U of A of all things, but I see how we can connect the dots potentially to bring in multiple people, and how important it is. Your comment about IP and how IP is now, in fact, a barrier. The inability of academics, we as academics don't really know how to partner with industry types and get our messages across. And I see that so often. And the potential of the basic science here is incredible, but it also speaks to the fundamental problem of animal models. And it's not just from, where you sit. I found it really interesting that your problem was the animal model was hard to work with, and you don't have a lot of animal models. We've had speakers in the past that have told us that apparently we've cured Alzheimer's 40 times already. Except it's been in mice, but it's not translatable to humans. So the animal model is great, works really well, easy to work with, you can solve it, but it doesn't actually translate. So all of these problems are problems that we as basic scientists, clinical scientists, translational scientists, and just people who want to improve healthcare really need to know about. And I want to thank you for being here with us today, because it's so important to hear about these, about the work you're doing and where we can collaborate. If there's anything that I'm sure any of us can do to help in any way in the future, please don't hesitate to reach out.
Oh no, thanks so much. That was a lot of fun. So thank you.
Oh. So thank you, everyone. I think this will be our last of these spotlight talks this summer, but do consider rejoining us in September, where we're already starting to plan our talks and interventions and panels for the next academic year. So I'd like everyone to clap virtually and keeping to time. We are just 2 minutes under the hour. So thank you very much.
Great. Thanks, everyone. Have a great summer.
Take care. Thank you
very much.